So we'll close with the most exciting of all, which is CAR T-cell therapy for myeloma targeting BCMA. There are many abstracts at the meeting looking at different CAR T structures, different ways of manufacturing, but we'll focus, I think, on the two leading targets here, which are from Celgene BB2121 and then from Janssen, the Legend LCAR. There are others too in clinical trials from Juno and other companies, but what do you think?
Well, I would say that all of us at this table would say this is the most important thing that's come through for myeloma in the last 20 years, no doubt about it. With BB2121, an extremely heavily pretreated population, between 7 and 23 prior lines of therapy, at the appropriate dose, more than 150 million cells, the response rate's 95% in this population. And the PFS is right around a year, so 12 months. These data were presented at ASCO by Newport, and everybody was just, I think, blown away with this data in such a heavily pretreated population. We're now getting data here. We're getting an update from the Legend data. This is the study that was done in China. And they've enrolled now over 70 patients in China. They're going to do an update of those data here, but it looks like in the abstract that the response rate is very similar, 88%. And really the benefit is in those people that achieve a CR and those people that achieved a CR, the PFS is 24 months. So this is 2 years of PFS in this group.
These are really heavily pretreated patients.
So in China, the heavily pretreated,
that's true.
It's 3 prior lines of therapy. I will say that we have the only U.S. patient that went to China and got this at Legend, and he is a legend for doing this, okay? And this gentleman was penta-refractory and went over there and we said, oh, this is your last chance of getting something that's going to have durable response. He's 15 months post this therapy now, and he's in remission, and he won't let us do a bone marrow biopsy to prove he's MRD negative, okay? Very important.
I was touched by that in the Legend update. The benefit is really for those patients who are able to get to that MRD negative response with that therapy. So the separation in the curve based on response, I think, was striking. So for certain patients, this is absolutely game-changing, but there are some patients that are not going to be
So there is a relapse
reached.
rate of, there is a PFS now, 50% PFS, so there are people relapsing, which initially we'd hoped we wouldn't see, but we are seeing those.
But I think, it was like 24 months in the responders, but 6 months if you did not get to that depth of response.
But those are your first trials, one dose and then wait. So as we get smarter, can you reactivate the immune system? Can you, are we going to be the checkpoints, immune modulation with IMiDs? Are we going to reinfuse? Are we going to, there's all sorts of little permutations that could happen. Plus, it's a highly competitive field. So I think there's going to be a lot of incentive to look at various strategies in this area.
Mm-hmm. Toxicity, Faith?
Yep, it's there. There's no doubt about that. And again, it's these unusual cytokine release syndromes that are.
They seem to be, we've sort of figured those out with tocilizumab, but the neurologic complaints we haven't figured out yet.
Yeah, no, and certainly there seems to be, kind of epilepsy and weird things that you're really not kind of.
Cerebral edema is the big one, right?
Yeah.
You've treated a lot of CAR Ts at San Francisco yet? Are you still in the early days? Or?
We've probably treated 20. And I am actually impressed on 2 things. One, some people don't respond at all, and they have no response whatsoever. And in others, a year later, again, they're MRD negative and don't require any other therapy. They haven't required any maintenance therapy, which is pretty impressive. I do think that we need biomarkers. We need to know more information about the microenvironment. I think this is where the microenvironment data is going to be so important of who's going to respond. And is there a way we can change the microenvironment before they get this therapy to make them a better responder to this?
What about timing of this? Will be my last question for the day here. Where is this going to end up? Right now we're treating end-stage patients. Where do you think this ends up?
I'm looking forward to learning how to use it better. To Rafael's point, this is very early on. It's a huge step, but it's the first step. I think we need to learn how to manage the toxicities en masse, ideally without steroids. We maybe can toy with the construct a little bit better. It would be great to get sustainability. So there's still some challenges and some room to go. once we get there,
You
moving
ducked
it
the
up front
question.
would be great.
You're ducking out on me. Is it going to replace transplant? Let's get to the heart of the matter here.
I think it could well do replace transplant. I'm particularly excited about those high-risk patients potentially up front, where we know that our current, it may only be a small percentage of patient, 10% or whatever, but we know our current therapies are not, good for them. I think that's a really great place
Raphael, I'm
to
going to
try.
give you the last word. We talked about 4 drug combinations, transplant, consolidation, years and years of maintenance, millions of dollars worth of therapy. Is this a solution for the economics of this as well?
Oh, it is. Let's not be, let's be not, what's the penny and pound foolish. I can't blanking on how you go with that saying, but if you think of your best regimen that you can induce MRD and potentially you do something like CAR T cell up front and then you do MRD testing and you're negative and we start getting the data, it turns out all this push for all this craziness is going to lead us actually to better economics, because you could envision in the best case scenario that we have a future where we treat myeloma like people treat 2-CDA, for cladribine, for hairy cell leukemia. So we can't stop right now because we're just at the brink of seeing even greater progress. And I'm going to say, I think we are going to see a significant number of myeloma patients being cured quite soon.
So when should our community colleagues that might be watching refer a patient for CAR T? Do they wait till they've exhausted, or should they be sending them in earlier?
I think during induction.
During induction?
Absolutely. Get them set up so that you can help them dictate their therapy pre-CAR T.
I mean, most of the local docs now, I think, treat patients with lenalidomide and bortezomib, and most of the patients eventually become double refractory. So I think when they're double refractory, that's a time to send them for some of these experimental therapies. And we have hopefully more trials opening up over the next year or two and more slots. The problem is we have plenty of patients for the trials right now. We don't have enough slots.
Yeah. Well, thank you, everybody. This has been extremely informative. Before we end this discussion, I'd like to get closing thoughts from each of our panelists. So, Dr. Martin, closing thoughts.
So my closing thoughts are that, for the treatment of the transplant ineligible patients, I'm going to start treating them early next year with lenalidomide, dexamethasone, and daratumumab. And when they have first relapse, which is 50 months down the road, they're going to get either a BiTE or a CAR at that point in time, and that's going to be the relapse. In the transplant eligible, they're going to get a 4-drug combination, a transplant and maintenance. But a year from now, they're going to get a 4-drug regimen and a CAR. That's my prediction.
Excellent. I feel like I'm compelled to go to the other extreme. I look forward to identifying patients and being able to personalize the therapy a little bit better. I think for some patients, absolutely, kitchen sink will be great. But if I could identify the patients that maybe don't need a quad up front, maybe don't need a transplant, or maybe if they have sustained MRD negativity, I could cut back on therapy. I look forward to that day. I don't know if it's quite in the near future.
So I'm going to close with the thought that something that's called the value of options. The value of options is a term that economists use to describe options that open for patients because they can live to the point that those options become true, which was what happened with AZT. Patients who lived long enough to see highly active antiretroviral therapy are now living among us. So for any patient who might be seeing this, if you have newly diagnosed myeloma, we have, in many cases, a possibility of controlling the disease for many years. And how we will treat myeloma in 2028, who knows? But I think the push is right there, and there's a lot of optimism in our field.
Faith, last words.
That's quite scary, isn't it? I think that one of the good things about this ASH is that we've discovered there's actually lots of options for patients, and it may be confusing trying to figure out which one to use, but to some extent I might argue that actually there may not be a wrong answer. There's actually many correct answers, and we also used to talk about patient choice. I think we're now actually in a position where we can choose the best regime for a patient.
Thank you. Thank you all for your contributions to this discussion. On behalf of our panel, we'd like to thank you for joining us, and we hope you have found this peer exchange discussion to be useful and informative.